Welcome everyone to our presentation. I am so excited that you want to take a bold leap, not just a step, in pathology. Welcome to the Muse sponsored talk. I'm going to be your host and I'm going to introduce all our esteemed panelists. Let's start with Dr. Levenson. Dr. Levenson is board certified in anatomic pathology and specializes in instrument and software development for medical applications. He is a professor in the Department of Pathology and Laboratory Medicine at UC Davis and a fellow of SPIE, which is a major optics society. He's a co-founder of MUSE Microscopy, focusing on advancing imaging technologies in pathology. His research involves mass tag-based multiplex ion beam imaging, AI for image analysis, and cancer immunotherapy. Previously, he served as vice president for research at Cambridge Research and Instrumentation, now part of Akoya Biosciences, where he led various innovative imaging developments. Notably, he recently published a project using pigeons to diagnose breast cancer in pathology and radiology images, which receives considerable attention. And at that time I was abroad and I did hear about the paper, and it actually was pretty famous. So welcome to the stage, Richard. Matt is the CEO and director of MUSE Microscopy. Welcome, Matt. And he has over 25 years of experience in technology and business. He specializes in biomarker discovery and development, earning recognition as an innovator in scientific circles.
As a co-founder of Liquid Biosciences, he focuses on advanced AI and bioinformatics.
Matt has established partnerships with institutions like the University of Pennsylvania and the Mayo Clinic, contributing to groundbreaking developments in oncology, autoimmune disease, and drug response diagnostic. He holds several US patents and has served on the boards of various companies, bringing extensive expertise in mechanical engineering and executive leadership. Welcome, Matt. Dr. Rao. Welcome, Dr. Rao. He is a tenured full professor at the David Griffin School of Medicine, UCLA, where he serves as the Vice Chair of Diagnostic Technology Innovation in the Department of Pathology and Laboratory Medicine. He is also the Chief of the Cytopathology Division and the Director of International Telepathology. His academic interests include investigating cancer biomarkers, nanomechanobiology of cancer cells, and digital AI applications in pathology diagnostics. Welcome, Dr. Rao again. Dr. Zhao. Dr. Zhao, we are not going in the order. Yes, Dr. Zhao is diagnostic and investigative pathologist with broad interest in cancer pathology. She was instrumental in making the studies mentioned in today's presentation a reality. Dr. Zhao is an innovative leader in the space of digital pathology in China, fostering international collaborations to increase the advancement of digital pathology for providing timely patient care, including the collaboration with UCLA. Dr. Jeff Edwards, welcome, Dr. Edwards, is an accomplished board-certified veterinary pathologist with over 35 years of experience in the field, with 25 years at North America's largest veterinary diagnostic reference laboratory, where he served as chief of anatomic pathology. In 1979, summa cum laude graduate of Texas A&M University School of Veterinary Medicine with fellowship in comparative pathology and an MPH from Johns Hopkins, Dr. Edwards has continually driven innovation in veterinary pathology, notably spearheading the first global transition to 100% digital pathology and integrating AI for enhanced cancer prognostication in animals. Currently, he oversees clinical and technical operations at SmartPath, leveraging disruptive technologies to transform how pathology and laboratory services are delivered. Dr. Chandra, welcome to the panel. Dr. Chandra is the Senior Vice President and Chief Genomics Officer at Path Group. He completed his medical training at the New York College of Osteopathic Medicine and underwent residency at NYU Langone Medical Center. Dr. Chandra holds multiple board certifications, including anatomic pathology, clinical pathology, hematopathology, and molecular genetic pathology. He received fellowship training in oncological, surgical, and molecular genetic pathology at MD Anderson Cancer Center and in hematopathology at NYU Langone. And Accomplished researcher, he has published numerous peer-reviewed articles and presented at various pathology meetings, including this one right now, as a member of several professional organizations, including the American Society of Clinical Pathology and the American Association of Molecular Pathology. Dr. Chandra is recognized as an international thought leader in precision diagnostics and cancer genomics. And last but not least, yours truly, Dr. Alexandra Zhurev. I am going to be your host. I am a board-certified veterinary pathologist, practicing toxicologic pathologist, and you might have seen my online platform, online educational platform about digital pathology place. Welcome to this presentation. Welcome all panelists. I'm going to ask you to have a seat in the front row. And our first speaker is Dr. Richard Levenson.
Good evening. This is a real pleasure for me. It's awfully wonderful to see something that emerged as a little box in our laboratory grow into this fantastic offering, which I think will transform pathology. And to that end, what I won't do is take any content away from the people who are going to talk about all of the wonderful diagnostic applications of MUSE, but instead I'll talk a little bit about how it originated, and then I want to just follow my own amusement and show you that not only is it good for standard histology, but it also opens opportunities for really new discoveries. And I just was reminded of an old saying, people thought that new science led to new technologies, but it's actually new technologies lead to new science. And I think over the years we're going to discover all kinds of new kinds of pathology as we explore not only the sort of standard H&E-like capabilities, but also what we can do. And I'm going to show you en face imaging. And there's one of the pigeons. So just a little bit of perspective, pathology as a field has evolved very slowly until the last few years. This is an image of a Van Leeuwenhoek microscope. It was developed in the 17th century. And these are images taken by a modern version of it. And you can actually see red blood cells and white blood cells. They look a little vague because there was no stain. But optically, that microscope would still be sufficient to sign out cases right now. So that's where we are. One of the other things is, of course, the potential for slide-free imaging to greatly accelerate even standard pathology. And on the left of the slide shows our state-of-the-art biopsy facility at UC Davis. And then, as you all know, after the biopsy is taken, it has to go through all of these steps, the formalin, the wax, the slicing, the floating on slides, the staining, and that's what a pathologist sees. And for it to become digital, it needs yet another step of whole slide scanning. And all of that takes hours to days to weeks to never, depending on where the laboratory is relative to where the patient is. And that's certainly true in global environments where path laboratories, histology laboratories might be hundreds of miles away from where the patient is. So we developed in our lab what's called MUSE, microscopy with ultraviolet surface excitation. And it was published a few years ago. And what really distinguishes this box from a lot of the other very, very capable opportunities out there, which include two-photon, multi-photon, other OCT, other slide-free microscopies, is how simple it is. That means it's robust and very, very reliable. And so, but here's how it works. How does MUSE work? It works because ultraviolet light at 280 nanometers doesn't go into the tissue very far, just a few tens of microns at most. So on the top, you see an image of kidney taken at 405, which is blue light, visible range, and you can see something is there, but it's blurry. This exact same field is shown on the right. If you just change the excitation pathway to or domain to UV, 280 nanometer, and suddenly all of those blurry features suddenly pop out as high-resolution surface. So that's all in black and white. But the other great thing about MUSE is that with a single stain solution, you generate not only sort of interesting images, but multicolor. So while the images that other people will show you can be converted into very, very convincing H&Es, I, as a sort of sort of advanced dilettante, I'm very interested in all of the extra colors and trying to correlate what that's trying to show us. So what you can see here is a blood vessel, and with that single stain and a single image, you can see the internal elastic lamina, the external elastic lamina. You can actually see down into the vessel because it's not a thin 5-micron slice, it's a solid piece of tissue, which means that we can see really much more surface features. If the surface is going up and down or doing something interesting, we will see that. And not shown in my talk, but some of the features that are just below the surface that are continuous, like blood vessels, show up in MUSE. If you make an H&E slide of those, they completely disappear and show up as little circles of capillaries that don't connect at all. So why is it useful? Well, first of all, let me show you this. It's a challenge to the audience. What's the diagnosis? It's Italian sausage. This is why I became a vegetarian. You can see nerves and arteries and veins, and I thought, I'm going to stick to tofu from now on. So that's useful. And then one of my talks is also the things that you can do you can't do with standard slides, which is we can see images in the opposite, in a plane that we don't normally see on a slide because we have to slice it at 5 microns thick. And to see anything en face, which means how you look at things, you need to align what you're looking at exactly within that 5 micron thick thing. And if you're out of that plane, it basically goes off the slide. You don't see it. So on the left is more or less what we see standardly. This is a stomach, and you can see the glands and the crypts going down into the vessel. That's a very familiar image. But if you wanted to see how the stomach surface looks like, that would be an en face plane, and that's shown on the right. So that's what we can do. So I'm going to show you some examples of that. And I think, although it's really early, and because I'm lazy and haven't done the work, I predict that there will be new pathologies coming out of these new image modalities. So here's something, this is just a standard H&E slide of kidney artery, or a small vessel in the kidney. And then this is a very similar vessel image with MUSE, also in the kidney. And what you can see there is the muscular surface. You can see the epithelial cells going down. So this is the topography, the sort of semi-3D or 2.5D enhanced imaging that we see with MUSE. So there are two things that really make MUSE sort of different from H&E. One is, well, additional colors, also this more surface information, and of course the speed. So these are just more pretty pictures to give you an idea of what's out there. These are blood vessels in breast. So did you know that blood vessels actually make continuous structures? They do, and you can see them both on top of the fat and actually even below the fat, because fat is semi-transparent. And there's a nerve up at the top, which I won't take time to show you. Collagen, it's an important feature of our tissue, if you will. I'm sure you would agree with that. So this is a canine mast cell tumor. It's a tumor that killed one of our dogs, unfortunately. And the blue cells are the tumor cells, and the pink stuff is the collagen. And you say, okay, there's collagen in this tumor. But do you really know what collagen looks like? Collagen in a mast cell tumor. And the point about this is it looks sort of semi-3D, but it's a single image. It's not 3D. You don't have to spend a lot of time cruising back and forth in a giant three-dimensional data set. It's all here, extra information, but not, I would say, not too much information, which is really exciting. Here's an image that, or this is an image pair that I put online in LinkedIn, and got a lot of people responded and said, blew my mind. Schwannoma, right? Standard H&E. Is that what a schwannoma really looks like? I would suggest that it really looks like that. Now, we can, with MUSE, make an image that looks just like the image on the left. It's just how you cut it with a razor blade. It would look like that. But if you leave the surface more or less intact, you can see really how the strands of the tumor cells intertwine and create this 3D structure. Choroid plexus, do you recognize that? It's sort of a standard-y thing, but is that what choroid plexus really looks like? Try that. That's the en face image of a choroid plexus, where you don't slice all of those structures into floating islands. It's not a floating island. These are continuous, highly productive, making CSF things. Endocardium, you've heard of it. What does it look like? I challenge you. So up there on the left at the very top, a little E up there at the top, it says E for endocardium. Most people see it when you cut through the heart. All you see is this tiny little cellular layer. But what would happen if you cut a heart open and then image the face of the interior of the heart itself? That's what it looks like. There are the muscle cells, and up there on the top left corner are the endocardial cells as a sheet, lining and sort of covering the system. That's very important structure because it protects the muscle cells from a lot of the things in the heart. And I'd never seen it before, and I doubt if any of you had either. So one more example, very similar example. This is a peripheral nerve with perineurium, which is basically the same as the endocardium. It's that sort of, you can sort of see it down there, perineurium, and a little arrow pointing to that thin layer at the bottom. Again, all you're seeing is one cell at a time. Well, this is an image of a peripheral nerve, a major nerve. You can see all lovely axons in there. But you can also see on the left, it's a little vague, but you can see the perineurium as a complete sheet. Again, diagnostic, absolutely not, but informative, absolutely. And presumably, if you start looking at things in disease situations, you may see new science, new pathology. And here's one indication that I think is pretty cool with something that I've never seen before in my life and I don't think has ever been described. This is lung, semi-3D, because lung, as you probably knew, are not just white circles on a slide. It's actually 3D alveoli, and you're sort of getting a sense of that here. Those orange cells in the middle are histiocytes. And what you can see, I don't know if this arrow is showing up, not really. What you can see is that big cluster at the bottom is connected by a thin blue tube to a tiny cluster at the top, and that tube is sort of encapsulating the little stuff, and it seems to be going around the big as well. That tube is not a vessel, it's not a lymphatic, it's got no walls, not cellular things in the wall. I don't know what it is. I think it's so fragile that it gets completely destroyed if you try to make a slide. It's never been described to my knowledge, and again, we have just so much more to learn from this otherwise highly productive diagnostic tool that's going to change standard pathology. So another example of en-face imaging is here. Actually, en-face imaging is literally a photography term for meaning taking faces of people and as you look straight at it. So the guy at the top is the one who really developed and built it, and he left us to go to Emory just recently. And last, to give you some confidence in the AI opportunities that this is also going to provide. So I asked DALL-E, which takes text and turns it into images. I said, please give me a photo of pathologists swimming in a bowl of soup. Are you ready? No. So I will stop there, and thank you for your attention.
Thank you so much, Richard. So all these images are created in an instrument that MUSE Microscopy has built. So I welcome Matt, the CEO of MUSE Microscopy, to the stage to tell us a little bit about how this is happening in this instrument. We might need a...
Okay, there we go. So I'll walk you through, this is SmartPath, this is the future of digital pathology. And this is a surgically gross tissue being directly dropped into a cassette. It can be up to 5 microns thick, or 5 millimeters thick, I'm sorry, 500 microns. It's coming under a UV array, non-visible light, and we are exciting the tissue. And what you can see is that what Richard's demonstrated is these beautiful colors and imagery that we don't yet know about. We've come up with an elegant solution that in less than 10 minutes will produce a histology-free, slide-free solution to potentially preliminary diagnosis. And we think that the data in the UV realm is the kind of frontier to be explored going forward. So I wanted to thank everybody. I was going to do that second, but I want to thank everybody for joining us tonight. We're excited as MUSE, as a company, to be here to show all of our colleagues what is possible. Dr. Rao and some of our guests that are here have been using it clinically. And, you know, I think that the time has come where, considering this to be digital is a thing of the past. When we can take a fresh or formalin-fixed piece of tissue, leave its integrity entirely in place, while being able to investigate and get what we need, which is diagnosis in an H&E slide that is familiar to all of you in the room and all of you online, but preserve and bank the data that is, I think, the frontier going forward. And we'll talk about that more on our panel. The data richness in the spectral realm and in the spatial realms using MUSE is not only low cost, but it builds the bridge between the pathology diagnosis and that patient, whether eventually where we believe we want to be, which is outpatient, points of care, all the way to the surgical suite, the intraoperative space in veterinary medicine, or where we think this really has legs to excel in further research among a lot of you here and a lot of you online, which is in the biopharma space and also in the research setting. So we want to thank everybody for coming. We're excited to share with you. Please stop by the booth and please do engage with us on the open questions.
Thank you so much, Matt, for showing this. The booth number, if you don't have time, well, today it's closed, but 528, and we have the technology, we have the instrument. Where's the data? And our next presenter, Dr. Rao, is gonna talk about a collaboration that he led that generated a lot of data to show you how this looks with real-life specimen. Welcome to the stage, Dr. Rao.
Thank you. Well, I really have a lot of respect to Richard, who pioneered this technique. I've been working with industry at different settings, but I have to say this is the project that excites me so much. And I've been privileged to work with people like Matt and Paul and the entire MUSE team. I have witnessed, how they transform the concept which Richard developed into an actual instrument that eventually I think it will make a pathologist's life easier, right? And also transform the pathology practice. So with that, I'm going to, again, thank Richard for laying such a great foundation for us and to make my life easier too. So I'm also wanted to just start to thank my collaborator from Second Affiliate Hospital of Zhejiang University, Dr. Zhou Hong. And I have to say, even though we've been working together for 15 years, almost, telepathology with this hospital that we consider to be a strategic partner. But in the last 5 years, I think, we really worked on this together. So I want to thank her for joining us on this effort. I've been working with industry for quite a few settings, like I mentioned, but with the MUSE, I'm only working as an advisor for now. Maybe at some point you give me a job, Matt. So, we've been so much going on with the digital and technology in pathology nowadays. Of course, one is molecular diagnostics, but from the digital technology perspective, think about it. We are still based on slide, right? So it's still the very traditional way of getting materials to cut, put in formalin, and embedding and cutting and staining. And this is really has been the foundation. And nothing has been changed, even with the AI and all these things. So I think this is the time now to think about new ways to make a diagnosis. And thanks again, MUSE, for making that happen. I think I don't want to spend too much time, because already we learned about the basics of the MUSE. The entire process is so easy for us, but on the other hand, if all I got is just a fluorescence image, as a pathologist, which I do, quite a few biopsies, I don't want to see the, fluorescence slide. I want to see HE slide, right? Because otherwise I have to train myself again. So that's not a good idea. So I'm so glad that, MUSE can make this fluorescent image converted into the regular HE so I can see it. So the concept is there, and we have the machine. The question is, how can we generate enough data and confidence? Of course, we have to go through the regulatory things eventually. But even before that, we need to know if this really works on human tissue. We know we worked on the animal tissue, right, at some point, but we needed some human studies. So I wanted to share with you the first project that we did on the breast tissue. So you may ask, why do you do that in China, right? We have so many breast cancer here. But I'm not sure in your institution, but in mine, we don't really have frozens anymore, right? For this kind of projects, you need a fresh tissue, you need the formalin-fixed, and you don't want to impact, the process downstream. So the best thing to do is to get a frozen tissue, for the frozen section diagnosis. And that's why we do that in second affiliate. And if you ask Dr. Zhou how many frozens they do per day, 180. But, that's just not possible in at least in my institutions. So we did like 2 phases. The first is the initial testing, which is basically take the instruments and put the tissue in and see what we got from there. And so it's not much a controlled, parameters. And this is what happens. We basically stuck in the frozen section room, and Matt and the team worked so hard, and the guy who had coffee is relaxing. I'm just watching and see what happens. We really had a good time together. And it's very efficient because we managed to get a lot of data in typically about 2 to 3 days, trip. So the first project, we, want to try both the fresh and formalin-fixed tissues. And also, most of them will undergo frozen section diagnosis. So we know what is the diagnosis at the time. And then we process the tissue. So typically we have 3 images. One is the Muse image, which we call RHE. And then second is something we call recovered. So in other words, after we scan the tissue, we put the tissue into formalin and processing like just the usual tissue. So that's what we call the recovered HE. And finally, we have a regular HE, which is the Miller tissue fragments taken from the Miller tissue that we use for Muse scan and process like usual. So we have GHE, which is the glass, the regular HE. The first phase, we tested about 27 samples from 19 patients, including 16 with malignant diagnosis, including DCIS and above. One of them, which I will show you, is I have a hard time to tell what it is. The frozen section also was not definitive. And we also have 10 benign disease. And that's another important reason we do that in China, because even the fibroadenomas, fibrocystic, tubular adenoma, everything they will do frozen. So this is very nice, so resources for us. And this is what we got. So on the left side, you can see the native image, which is the fluorescence image. Again, I don't want to make a diagnosis based on that. Sorry, Richard. It's just beyond me, even though I've done a lot of research on fluorescence. But the virtual HE is the initial HE, which is not the optimum, but I have to say right now, the image is much better. And the recovered HE and also the regular HE. So, and if you look at the normal, benign, and malignant, it's very easy to tell. You don't have to relearn your pathology. Fibroadenoma is look like fibroadenoma, right? And this is very easy to make a diagnosis. And this is one of the cases that we just this is the cancer malignant case, which you have a lot of infiltrative glands in between the fat. And this is the case that really we have problem. I didn't know at the time of the frozen what I'm looking at, because looked like there was quite a bit few giant cells. But there's like an infiltrative border in the tumor. So initially I thought this could be some kind of treatment after chemo, maybe histiocytic reaction or sort of like that. But it turned out to be, I think Dr. Zhou told me, actually it's a sarcomatoid metaplastic carcinoma. So I think I had a hard time to make a call on the frozen and on the Muse image as well. So that's the first phase that we actually went couple of times, right? And then the second phase, we said we wanted to go further and now do some actual simulating studies that really to test the concordance of the Muse interpretation versus the regular HE. So for this study, actually we went there for how many days? 2.5 days, right? So we managed to get 38 tissue samples, including 22 formalin-fixed and 16 fresh tissues from 28 patients total. I read the images myself, but I have to say I'm not a breast pathologist. I do mostly GYN and GU and cytology. So it's a little bit hard for me in terms of making a call. But for this particular project, we are not comparing specific diagnosis. I'm just giving you a ballpark, benign, borderline, or equivocal, or malignant. So we use the past presenter to read the image. And we do have 2 weeks, like a washout period between the Muse imaging reading and the regular HE. Okay, so this is actual virtual image that you are going to get with the Muse scan. And I think you can probably easy to tell, I don't know if I can enlarge or, you can see the tumor cells, right? And you can even find mitosis. It's very easy. But really what is fascinating me is this thing here. I don't know if you can see it on the low end. Where's my arrow? Okay, it's not a, so let me just skip to the next slide. So this is the pathology diagnosis we got from Dr. Zhou. And notice that this is, I don't know which translation language you use, AI, Google, or whatever. It's pretty good, but actually, one thing I wanted you to pay attention to is that it says vascular invasion is negative, but there's one lymph node positive. But look at this. That's actually the image that I showed you on the low end. You can see the vascular space here, right? And here's the nice tumor cells within the vessel. Look at the elastin. Remember what I told you, the final diagnosis? The lymph node is positive, but the actual lymphovascular invasion was not thin. Because on the Muse, it's so easy to see it. Okay, so that's the point that I think is very important. I think Richard mentioned that. You can see the vascular structures very well. And I can guarantee you, if you have this on the Muse, you can call lymphovascular positive. It's probably not the lymphatics to me, but I think this is what I'm trying to show you. So overall, we compare the fresh versus fixed tissue and see how quality of the imaging compares between the two. It looks very similar. So in other words, the fresh tissue and formalin-fixed, that doesn't make any difference in terms of imaging quality. And overall concordance is not 100% yet. We still have some discordance. Like there's one case I call benign, but it turned out to be malignant. That has to do with the area that we see on the actual regular on the image, the covered image was not actually present at the time of just the sampling issue in a way. Okay. So if you look at the overall concordance between the Muse and the final diagnosis, I mean the entire tissue, based on entire tissue, it's still more over 85%. So that's pretty reasonable to me. So that's one project that we did in China. The second project really we wanted to demonstrate is to test if the Muse fixed tissue, processed tissue generate good enough DNA and RNA quality for downstream molecular study. We're not doing any actual point mutation or sequencing, but just look at the DNA RNA quality. Because what we really wanted to demonstrate is that Muse actually, because it doesn't involve in formalin fixation and pathogen embedding, it may produce better quality DNA and RNA. So that's what we try to demonstrate. And this is what done on the prostate, because we have prostate spore at UCLA. So we don't have to go to China for this, and we decided to just do that at the UCLA. So in this study, we really just wanted to look at the DNA RNA quality between the Muse processed tissue versus the formalin fixed. So total about 28 samples from 14 patients, and we compare that to formalin-fixed in about 22 tissue and from 7 prostatectomy specimens. And I don't want to go through this to you with very much detail, but just in general, the MUSE scan, both RNA and DNA generate better fragments compared to FFPE, but this is what really makes the difference. If you look at the top bar chart, and if you compare the MUSE with the formalin-fixed tissues, the big difference here is the DV200. I'm sure you know what that means. It's just the DV, the variance, the size of the nucleotides that are over 200 for RNA and over 1,000 for DNA. It's significantly better with MUSE processed tissue compared to FFPE. However, the ring number doesn't show much difference. I don't know if that's, we are actually doing more tissues now. I think we look slightly better, but I think that's not a major difference that we see here. The major difference is mostly at the DV200, which is really the fragments that are large enough for next-gen sequencing. So I just wanted to go quickly to you about what we have done so far, and has been a long time in coming. And I'm glad that since I'm moving much quicker now. But from what we learned is that the direct-to-digital on both formalin-fixed and fresh tissue generate about equal quality. I think doesn't make too much difference for the MUSE scan per se. The second thing is that we can see at least 90% concordance if you try to give a benign to malignant classifications of the tissue on the breast. So that's not a problem to me, but I think I want to get to probably more than 90%. So Matt, you still have work to do. Okay, I need more than 90%. Okay. The other thing is that as we expected, the MUSE scan tissue generates much higher quality DNA RNA in terms of the size of the fragments. Of course, whether that really changes the molecular diagnosis, I think Preneur will have to tell me maybe 2 years later, right? Because we have to do a lot of work on that. The process is very quick, okay? And I still have to get used to some of the images that I see, which is, again, it's not a fluorescence, it's like simulating HE, but the cellularity is a little bit more than usually what we see on the HE section. So I keep telling Matt, don't give me too many cells in there. I want to see much thinner layers, which I think technically speaking is possible, right? So, and I have many people to thank, including people from my UCLA team, and there's a lot of leadership from my department, especially Sarah Joy, who really provided a lot of support for this kind of innovative projects in my institution. I also wanted to thank Dr. Zhou and her entire department, and the MUSE people, which I can't say enough. You guys really fantastic. But of course, the regular HE is one thing, but also I learned something else, is how to drink tequila, right? And Maltai too. So we really have a good time in China other than spend time in the frozen section area. Thank you so much for your attention.
Thank you so much, Rao. Now, I get to ask questions, and after that, you get to ask questions. So I would like to welcome my panelists back to your seats on the stage, and I'm gonna ask you some questions. Because now we know the background of the technology, we've seen the device, we've seen real-life data. I wanna know what implications this technology is gonna have. So the first question has two parts. It's the patient-centric question, and I'm gonna start with you, Pranil. With today's growing emphasis on rapid patient diagnosis, what immediate and future workflow enhancements does MUSE provide?
Thank you. That's a very good question, and let me answer that in a couple of parts. With the technology that you all see here today, you have the opportunity for rapid diagnosis. And so that's a diagnosis of a benign lesion or a malignant lesion in a very rapid format compared with the traditional pathology workflow. One of my colleagues who's in the audience who trained at Mayo Clinic, this was how patients were diagnosed, was through rapid frozen section diagnosis. And I will tell you, there is significant value in being able to render rapid malignant and rapid benign diagnoses. The real value, however, and just to back up a little bit, is I'm part of a practice where we diagnose about 150,000 solid tumor cancers on an annual basis. And we have to have good data for downstream molecular testing, good tissue, as well as good data. And so when you're able to make a rapid diagnosis and preserve certain types of tissue, like lung cancer tissue, for downstream molecular testing, that's a big win for the patients. And the third area of benefit, and then I promise I'll stop, because there's a long list of benefits with this technology, is the data that's gleaned from the beautiful images that Dr. Levenson showed. While it's not 3D, you do get topographical information, and you've got information about blood vessels as well as tumor infiltrating lymphocytes, and really the architecture around cells and around malignant cells. And today we are gleaning digital pathology, artificial intelligence, computational pathology algorithms from images scanned from an H&E slide. And you can just imagine the richness of the data that you can glean from the tissue through this instrument. So hopefully that gives you a good smattering of some of the advantages and the value adds to your question.
Mm-hmm. And the second part of this question is actually for Dr. Zhao. How will this technology you practice in China, how will it transform access to pathology expertise in your country and in other parts of the world, from primary care facilities to specialized reference laboratories?
Okay, thank you. I think in United States, they have a lot of pathology doctor. But in China, in large city, also enough pathology doctor, but in remote area, I think it's lack a good pathology doctor. So I think on one side, this technical can help the remote area. For please think about in clinical, one tissue get it directly and directly get imaging and transform imaging from internet to the central hospital doctor, and we can diagnose it and directly tell them the conclusion. So I think it's very helpful for the remote region hospital and the patient.
Mm-hmm. Thank you so much. And obviously this is your experience from China, but this applies to US, Canada, and all the remote areas. So thank you very much. And my next question is the dimensional advantage question. And this one goes to Richard, because you're a fan of high dimensionality of those images. So traditional pathology has been limited to flat 2D perspectives for over a century, and it still is enough to get a very accurate diagnosis. So how does the multi-dimensional capability of MUSE image unlock additional insights that are not accessible with standard approaches? And if you can share a specific example, that would be fantastic.
I'll share 1.5 examples because I want to comment about the lymphovascular invasion as well. So first of all, multidimensional can apply to spectral, that is to say color, as well as actual physical. And I've already shown you a lot of the physical information where we can see how surfaces are put together that you really cannot see with other techniques. But there's also, and I think I mentioned this as well, the fact that we're looking at an intact piece of tissue means that we can see things that are continuous in real life, but if you section them at 5 microns, they become just discontinuous points. And I've seen this with blood vessels, especially small blood vessels, where all you see is little point, with the H&E. And if you look on slide-free images, you can see them actually extend at great length and interconnect, and it opens up a whole new world for analyzing basically the tumor vasculature, which is obviously very different than normal vasculature. And just the last word about the lymphovascular invasion, which we heard about and saw a nice example. I'd point out that it's not just the fact that it's thicker and you can see down, it's the fact that the piece of MUSE tissue is actually thick. And the problem is with vascular invasion is that the tumor embolus, if you will, is not connected to the wall of the vessel. It's just sitting in there. And if you're not careful when you make a slide, that little tiny piece of tissue just falls out. And so I have a paired, not here, but I have a paired section of slide where there's a beautiful image of the H&E where the vessel is empty, and the same vessel on MUSE is filled with this tumor. It's because it simply fell out. So I think it's going to change some of the math for lymphovascular invasion.
Mm-hmm. Thank you so much. So the next question goes to Rao. The tissue preparation question. There is a flexibility with MUSE to use both fresh and fixed tissues. This flexibility is a clear advantage in sample handling. So what significant differences, if any, have you observed in imaging quality and workflow efficiencies between fresh versus formalin-fixed samples? This is a question we often get at the booth, so let's get it out there.
Well, as I mentioned in my data, we really don't see a whole lot of imaging difference in terms of quality. But I think for the workflow, obviously, I think the advantage is that you can get a good MUSE image in both settings is very important in practice, because think about in some small clinics, they may not be able to put the tissue immediately into MUSE. They can still put it into formalin, and they can still ship to somewhere like in the PATH group and do the scanning. So I think that's a huge advantage. You have the flexibility. And I also wanted to add the point, that we discussed about lymphovascular invasion. I was so stunned to see the image that when I actually, I have to say, I forgot to mention.
Seeing is believing, right?
Yeah, the actual imaging on the fantastic looking machine, I always call this coffee machine. The screen is much better than past presenter. So don't tell this to past presenter group. I don't know how somehow when I think it's condensed, when the actual image get transferred to past presenter. But if you look at the original image, I think, I was totally blew up. It's so good quality, crystal clear. The nuclear features is so fantastic. I'm still not 100%, I have to say, the cytoplasmic quality is a little bit not there yet, but it's just amazing to see the vascular. It's just amazing. I never, typically as a pathology, we told our residents, don't look at the lymphovascular invasion inside of the tumor, right? You look at around the edge, because otherwise you can't tell. But this particular case is right there. It's just so amazing.
Thank you so much.
I just have one extra comment on that, which is there are obvious differences between fresh and fixed tissue. One is tissue firmness and how it behaves when you confront it with a razor blade. And so sometimes if you try to cut fresh tissue with a razor blade, the tissue sort of jump out of the way, whereas if it's fixed, your razor blade goes straight down. And that can be a difference that makes a difference. But apart from that, the image quality can be very comparable.
Thank you so
Aleks,
much.
can I add something real quick?
Yes.
As far as 3D, a specific example of how that was very effective, I call it 2.5D actually. I work for another company as well, and we're on the cusp of developing the first fungal vaccine for humans and animals for coccidioides mycosis, valley fever. The H&E, this was a feline from Arizona. You could tell by the reaction, it's probably going to be that. Had to do multiple sections to find it on the H&E. You've been through all that. Serial section, the GMS, PS staining. The MUSE image, since it was 2.5D, was thicker and deeper. There it was. No additional sectioning, no special stains. I gave that case too. We did an animal concordance study. I may be able to talk about it later, but all my study pathologists were able to nail that organism just from that one image.
Thank you so much. The next question is the future technologies question. So this one goes to Matt. As healthcare starts to embrace AI-enhanced diagnostic, how is MUSE positioning itself to support advanced applications beyond basic imaging, specifically multidimensional reconstruction, quantitative analysis, and computational pathology?
Thank you. I think that as a business model for us, it was important to build what is maximally possible at the image acquisition and then back down from there. And so I think what you're hearing right now from either clinicians like Dr. Rao, Dr. Zoe, Dr. Edwards, is that we're trying to find the sweet spot with generating so much clinical data, so much data in the image that we have to balance the transition for pathologist users to be able to make that transition to a highly topographical image set that is still rich with all of the spectral data still intact. It's an order of magnitude greater raw data that will be available for applications in AI. We all see this in our glass slide scanning capabilities with a lot of the companies that are in this building that are really focusing their efforts on AI and assisted diagnosis of complex mechanisms that may not just be morphologic in nature like the traditional pathology. And so we're trying to work with our clinicians to balance how much data we present in that H&E converted image, which is an empirically driven exchange of the fluorescent image to the H&E presentation. I've heard a couple of analogies where it's a simulated H&E or it's a kind of computer aided or generated. This is a mathematical, empirically driven, so it is measuring the fluorescent responses that we know are binding to the cytoplasm and nuclei, and we're converting them at subpixel precision across multiple planes in an X, Y, and Z to generate that imagery. And so when we have cytoplasmic detail that needs more evidence or more depth, it's a fine balance. And so we're trying to find where is that happy balance. And that's why we're so happy to be working with so many of you in the audience and here on site, because we feel fortunate to be able to have that balance is 95% the way there, I think, at this point. In terms of the quantitative analysis and computational pathology, there's no doubt that our system is probably, in my view, generating the most robust, multidimensional, both spectral and spatial data available. And we think that the tools are out there and will be coming even further. And so we think we're well positioned as a manufacturer of hardware to partner with those companies leading those charges with so much talent in those spaces. So we think that we're foundationally, the hub, and these are all going to play the spokes. The team has done an amazing job at creating a highly robust and efficient, repeatable process that we can deliver at any level. Molecular, pathology, all the downstream analysis, everything can be done in fluorescence, and we can build that bridge to the clinicians.
Mm-hmm. I want to ask Jeff about a reality check question. This kind of ties into your experience with going digital in a big institution. What challenges does MUSE anticipate in clinical and research adoption, and what technological improvements are on the horizon to address the common objections you've encountered from practitioners, from those who are actually diagnosing the MUSE images for your studies?
Thanks, Alex. That question is so complicated. I'm going to answer that question with a question. I want to ask the audience, first of all, again, I'm a veterinary pathologist. By the way, Aleks is as well. We're probably the only two at this entire meeting that are veterinary pathologists. So. How many of you have a pet? A dog, cat, horse? I'm going to include birds, fish, no. Snakes, yeah. Quite a few of you. And you know that it's, despite the minimal penetration of animal insurance, even if you have it, it only pays for very minor things. Annual physical, CBC, chemistry panel, maybe dentistry. So it's a cash-based system, and here's a perfect example. 10 days ago, my mom, who lives with us now with her dogs, one of her beloved dogs, Rocky, had silica stones, bladder stones, very uncommon in the dog. They couldn't be eliminated by diet alone, so she had to have surgery. $7,000. That's a lot of money. $7,000 out of her pocket. I'm amazed at how much people will pay for their pets. It has continued to amaze me through the years what they will pay. Yet, at some point, for any of you that have pets, there's a limit. And I'd say we're fortunate. I think in veterinary medicine, I feel fortunate that euthanasia is an option. And my mom had asked me at the time, I told the surgeon, I said, look, if there's any masses in that bladder, I want you to send them to me to one of the 2 labs I work for. It's going to be, told my mom it'll be 5 days before I get an answer. Here's the anxiety of waiting 5 days. Son, what if it's cancer? I said, well, Mom, it's going to be more money, more expense. You'll have to make a decision. With that preface, most of you probably don't know, at least for veterinary biologicals, vaccines, drugs, feed additives, they're required to have FDA approval. For things like whole slide imagers or imaging devices for whole slide images, it's not required in veterinary medicine, nor is that unit right there. It's not required. That allowed us to, in veterinary medicine, to reap the benefits of whole slide imaging much, much faster than in the human field. Back to the question. The intended use in veterinary medicine will be quite different than in human medicine. I'll talk about it a little bit later, but we're going to have point of care units. These units will be placed in veterinary hospitals. The challenge is going to be, especially for veterinary pathologists, probably like most of you, are pretty conservative in your career. You don't take changes lightly just because somebody says so. So it will require considerable exposure to these images, which they've never seen before, though the ones I've talked to have shown they're extremely excited. It's going to require paired imaging where the veterinarian will show the MUSE image to the pathologist, but then the veterinarian will also drop that tissue in formalin. We'll have to go to the reference laboratory to where it's going to be produced as a standard whole slide image, and those will be examined side by side, just as all of you have done with whole slide images compared to your microscope, back and forth, so you could get that confidence yourself that the whole slide image looked the same or reasonable facsimile to the microscope. The other, I think, growing pains are going to be the person doing the grossing will not be in the grossing lab, not be in the histology laboratory, in the reference labs. This is a sea change. It's going to be done at the hospitals. So those certified animal technicians or the veterinarian will need to be trained on how to trim that tissue and to follow the protocol for staining to produce the MUSE image.
What I'm going to do a little bit of a change because I want to give our audience the chance to ask questions. So there is a microphone in the back of this room here in the aisle. If anybody has a question, please go line. Whenever I see you there, I'm going to interrupt the panel questions, and give you a chance to ask yours. So feel free to think of your questions. Show me that you want to ask one, and I'm going to do one more panel, but super quick, okay? And that's going to be one for Pranil. The workforce evolution question. Disruptive innovations always reshape professional roles and responsibilities. How do you think MUSE will reshape the responsibilities of pathologists, pathology assistants, histotechnicians, and modern laboratory?
Well, thank you for that question. It's one that we should all give some thought to in light of the fact that histotechnologists and laboratory personnel are becoming harder and harder to recruit. I see a nod in the audience. It's a real challenge to hire good histotechnologists. The job market is tough, and there are so few of them. Well, now with a workflow that's enabled by the MUSE system, you can take a sample and let's say it's benign. A benign diagnosis can be rendered in a matter of hours, and then all of the downstream efforts, the histologic processing, the paraffin embedding, the cutting of the slide, all of that can be potentially obviated. You've given a diagnosis in a matter of hours, and you have now decreased the amount of labor and the amount of technical work that you would need to put into developing that slide. Now think about it. We have, most of our tissue is not malignant. Most of the tissue that we receive in the pathology department is in fact benign. So I'm not saying that this will put histotechnicians out of work, but you would be able to repurpose histotechnicians for other tasks and other responsibilities. So I think that's going to be a very significant and major impact of this technology.
So I just want to jump in because I got an email today, very fortuitously, from somebody I don't know, but he's a histotech at UC Davis, and he wrote to me. He said he's been exposed to sort of what slide-free imaging is. And so here's one paragraph. He said, with my extensive experience in embedding and sectioning, I'm wondering if these skills might become obsolete as slide-free histology takes hold. I'm very much open to pivoting early if necessary and would love guidance on how to transition my training and expertise to your platform. So people want to, jump on the ship.
Yes, thank you for that. And a quick comment on top of that before we go to the audience question. I posted about MUSE on LinkedIn, and I got the same discussion about histotechnicians. How will they transitioning? So this is something ongoing right now. And now we're gonna address questions from our in-live audience. The questions from the virtual audience will gonna be addressed after this meeting. So feel free to write them in the chat if you're dialed in, and we're gonna get to you after the meeting. So go ahead, please.
Hi there, Sayag Khatak, a molecular pathologist, NCI. So I have a question. So the examples that you showed, the images that you were showing, many of them have a lot of architectural variability in the fields that you're showing. So how does this image conversion from the original fantastic MUSE images work, and the diagnostic efficiency work when you're looking at very homogeneous tissue like hematological malignancies or ITCs in lymph node, micromets in lymph nodes?
So you're asking the architecture features of the MUSE, is that?
So the architectural features kind of are predominant, probably that helps in the diagnosis. What about homogeneous tissues where there's not a lot of architecture, like
good question.
Lymph node metastasis,
Right.
Something like that?
Actually, the homogeneous tissue are much easier. The heterogenic, the one that I typically see problem sometimes can be challenging is like when you have mixture of fat with the muscle, and sometimes that the imaging can be a little bit challenging. But if it's homogeneous, like muscle, liver, kidney, uterus, those are very nice, homogeneous imaging to see. Actually, the morphology is much clearer than the heterogenic type of tissue. So maybe the example I show is opposite, but actually when I look at it, then the homogeneous tissue are much easier to make a call.
Thank you so much.
My experience with approximately 12 years ago, again, I said we didn't have to have FDA approval for the scanners and the image viewers. And at the company I worked for, the largest veterinary reference laboratory in the world, we went all in 12 years ago or so. We were scanning thousands of slides per night at 40x. And that conversion went so smoothly. In fact, one of your own, Dr. Jared Schwartz, had given me the 4 basic fundamentals to follow. He said, if you follow these fundamentals, Jeff, you will succeed. And I was amazed at how rapidly and how quickly those pathologists fell in line, or should I say the word?
Adjusted.
Became acquainted with and confident with the whole slide images and didn't want those microscoped or the slides after about 2 months. I'm convinced from that experience, and it was the largest conversion that was ever attempted still to date in veterinary or human medicine on conversion of whole site image. I'm convinced from that experience that the pathologist, the veterinary pathologist, will also be able to make the conversion to evaluating MUSE images as well. It's so radical, and thank you for Dr. Levenson for inventing it. It's so radical, but it's so simple, and in a way. And from the concordance study that we did, the pathologists were able to do pattern recognitions quite quickly. I can show you a slide on that later if you'd like, with a time very similar to that for whole site images.
Mm-hmm. Thank you so much. Let's take the next audience question.
Kamal Khurana from upstate New York. I just have two questions. The first one is, this technology is not going to eliminate the need for preserving the paraffin-embedded tissue because you need ancillary techniques like immunohistochemistry. So we will still need to preserve we will still need to embed the tissue in formalin fixed blocks so that we can perform immunohistochemical techniques?
So my earlier comments were directed towards benign tissue. And if you have, for example, a skin tag, fibroepithelial polyp, there wouldn't be a need potentially to make a paraffin-embedded block and for all the downstream costs. However, for cancer tissue, there is absolutely a need to make that block. And the advantage of a technology like this, and for example, in lung cancer, is you have the ability to preserve some of that tissue for not only the downstream immunohistochemistry testing, but also the equally necessary molecular diagnostic, molecular oncology testing that needs to be done on that sample.
Yeah, but for like polyps and stuff, like fibroepithelial polyps, skin tags and all, you don't know what is in there till you actually examine it. So do you think we need to still continue with paraffin-embedded tissue for all tissue? Because you don't want to go in hindsight and say, oh, we never preserved it in formalin and we can't do any immunostains if there's a surprising cancer that shows up in those benign, apparently benign tissues.
Yeah, it's a good point. And whatever is eventually done in the pathology lab, that will need to be validated through studies. So we're sitting here and we're proposing what the workflow could look like. But in reality, there have to be the appropriate studies that will need to be done to validate whether this workflow can obviate the need for additional downstream workflows. So,
thank you.
I think going back to your immunostain question, that's actually one of the things we are going to look into. Remember, any kind of immunostains based on antibody on the formalin-fixed tissues, it typically requires quite a bit of antigen retrieval and all that, right? So, but I think I think Dr. Levenson has done some early work to show that it definitely can, immuno can be done on the MUSE scan tissue, including fish, right? The questions like, for example, HER2, and it involves so much clinical validations for determining the cutoff, right? So that kind of work probably has to be duplicated on the MUSE scan tissue, but it can be done. I think it's just that you don't have to be really putting tissue in the formalin in order to do the immunostain. Immunostain can perfectly be done on the MUSE scan tissue, but the question is whether that the reading can be different or not. We don't know yet. So that's something we have to do some work to.
And just a word, I believe that there are state laws that require tissue to be kept sort of intact in paraffin for at least 7 years. And that makes sense because 5 years from now there may be a whole new biomarker that it might be important to pull this out and test again. So it's somewhat a matter of state regulations. But the other thing is that's not a high-speed problem. You can put it into formalin and paraffin and then just let it sit there. So it doesn't have to be done in overnight.
Just from the company's perspective, I talked about the hub and the spokes. And I think that the company would take the position that the instrument, in terms of antibody tagging and things like that, there's a whole field of science evolving in fluorescent tagging capabilities and things like that. And so I think a lot of the workflows that we're talking about now that are maybe more laborious outside of storage can all be done almost contemporary, sequentially at the time of diagnosis if you desire to on system. And I think that's where we're excited as a company to start partnering with those that are in those fields. And we've had a tremendous amount of interest in those types of technologies. This is the enablement that makes that whole field of science possible. And I think that what you're talking about is IHC and FISH and things that we're doing in the current work form, but that's not the work form that'll exist 5 years from now or 10 years from now. This is the foundation that'll enable the entire field to change, and this should be at the front of it.
Real quick, one thing, in veterinary medicine, we're not required legally to make tissue blocks. So hopefully, if a diagnosis can be made at the point of care, the veterinarian can just plop that tissue that was just imaged into formalin, leave it at his or her clinic, and that's the end of it. If we need it, we can always call it in with a courier, but no blocks have to be made.
The second question I have is, do you guys have any experience with the cytology stains or with the cytology smears, especially DiffQuick and all, or is there a potential for this technique to do DiffQuick staining on smears too? Because for rapid on-site, we need that. So
Not
something,
yet.
not yet.
I know Matt says yes.
It's technically feasible to do cytology, but I think there's a lot of very good solutions for rapid diagnosis in cytology. So I would argue if there was ever a desire to have a two-in-one offering, it's possible. It's not the focus of MUSE today.
Thank you so much. Let's take the next audience question.
Hi, I'm Trang Lally. I'm from the state of Washington, practicing pathologist. I have two questions as well. One, I'm pretty excited because I feel like this could be a game changer for intraoperative consultations, especially because there's a freeze artifact and cutting frozen tissue. It seems like this will make this really clear. Do you think the technology will be fast enough to meet the 20-minute limit for like the intraoperative consultation recommendations?
So from the company's perspective, and I know clinically, having worked with Dr. Rao and Dr. Zo's teams, that we're every bit as capable as the frozen section rooms. Obviously, not all frozen sections are created equal, but I would also tell you that the capabilities of having even faster turnaround time is there. It depends from a computational perspective whether we've built the system so that all the computation happens on board. There are capabilities and things that are in the works that as we go through the regulatory environment, and as we change the intended uses, and as veterinary pathology and research-only applications desire the use, we will have and do have solutions for cloud-based compute where the system is doing just image acquisition. That would allow you to do WSI imagery at full resolution with rendering in less than 5 minutes.
That's great because like you can integrate it with your digital like environment and have multiple people on consultation. If like one person couldn't read it, they can show it around instantaneously, right? Like that's kind of like the goal of what MUSE can provide, like instant imaging without all the artifact of cutting and not being able to cut a lymph node, maybe, that's gone. You can all have like whole slide imaging and instant access.
Yeah, for MUSE, the approach has been to create a box that does two things. It preserves the integrity of the tissue to the maximum ability possible for what is currently available in the market and for what can be in the future. So long-term partnerships with innovation partners in the building and online that will further the other new areas of science that don't exist today. And then secondly, the time and efficiency, the capital constraints and the workflow constraints from the, not just workflow in terms of footprint in size and in need, but also, as we all know, there's going to be, it probably is huge shortages in histotechnologists and pathologists both. And so anything that we can do to remove those roadblocks from their workflows and make it an aid that they can reliably work on quickly is what we're really aiming to do as a manufacturer.
My second question is like, because it's based on light imaging, can you in a way Z-stack your tissue? So you know how we always cut levels to find something. Is that something you can on Muse? It's like pretend like you have a TA and you have to cut levels and it's not on the first level. How does that work with Muse?
Yeah, so we take a extended Z-stack. You know, we're talking 20 and 25 microns of depth. And if you add the topography of the tissue and that, we could be sampling 70 or 80 microns, 100 microns even, depending on the tissue physiology. But I think that parsing the Z-stack layers is completely doable today. We can render those in, you know, we do render them in all in-focus imagery today. But if you wanted to take analysis and see that interconnectivity in cellular morphology at the various levels, not only can we do that from a locational standpoint on the data side, but we can look at any of the features physiologically or otherwise independent of any other feature. So you can look at cytoplasm of the entire tissue independently of any of the nuclei or any of the locations or clusters or any of the features about that. And so that's where we're really committed to that high data resolution and being able to parse that data in ways that are going to be meaningful over time.
So there's another way of looking at this, which is you have the opportunity to see whether or not your first view was in fact diagnostic. You see the cancer right there. You don't need multiple depths. And if you want to get really good true depths, what you take, for example, if you had a needle biopsy that looks like that, you would image the surface. But if you're not clear on what you're seeing, you could then cut into little sort of rings, flip them on their side, and image them, and then you would have a complete view through the entire depth of the biopsy all the way down its length. So that's actually better than serial sections because you would not be skipping really any major areas.
Thank you.
Thank you so much. Let's take the next question.
A very stimulating discussion, actually. I was looking into your presentations. I was wondering about the spatial resolution on the sections, because basically what I have seen here and what you have presented is what I would call low power pictures or magnifications. So my diagnostic work will be more related to pattern analysis. So I wonder actually, can you promise something with respect to more cytological details here?
I can go first, but you probably can get. That's a great question. And you know, even though I've tried to show the low power, but actually the images are almost like 20x. So you can see enough cytology, including cytoplasmic granules, and mitosis very easy to see, and apoptosis, and those can be very easy to see. And sometimes the nuclear membranes even can be seen. I think right now is that 5x scanning, 10x scanning.
10x.
But the actual imaging that I typically analyze, low power shot, is just try to take the original imaging, just put that there. So look like a low power, like 2x, but actually it's 10x, is right. So
one thing I'd just add, not to get too technical for audience, but if you had sampling at a 10x with a high numerical aperture versus a 20x with comparable numerical aperture, that's spatial resolution. That's one issue. But then you have spectral resolution, which is another. And I would argue we're capturing at least 2x the spatial or the spectral data, and bringing it all into a usable, what I would say, simulated 20 or 40x H&E equivalent is completely possible today. I think Rao and if any of the viewers that we've used would probably tell you, and I don't know, Jeff, if you have any opinions as a practicing pathologist, but 20 and 40x magnification is not an issue. And I think that the spatial resolution of the instrument today is on par with most white light scanners, because you benefit largely from the, I think, non-visible light brings the detail, right? So Richard's got publications at 400 nanometers. You can't see things using that type of light that you can at a short wavelength. And so when you couple those things, it's not just a Nyquist sampling that we're looking at, it's this other broader subset. When you bring them all together, we actually have improved, I think, image quality.
Okay, thank you very much.
Thank you so much. We do have 5 more minutes, so I would like to ask one more panel question, if that's okay with you. I'm going to skip the speed comparison question because we already covered this. Oh, we do have an audience question. Okay, let's do it.
Sorry for the last minute. So
no problem.
Yeah, it's fantastic technology. I was amazed that you guys put out all this effort. So I have a question. This seems like, to me, my understanding, it looks like a confocal, if I understand it correctly, it's like confocal technology on the live tissue. You have like all different levels, but you have you have ultraviolet. What's that? The preconditioning of the tissue. But what I'm thinking of this. Previously some group then doing the intravital microscope. So I'm thinking this technology that can be applicable to those kind of a scenario that you put a scope in, and you can scan it and have all the ultraviolet and to scan in the tissue and have all this optical data images sent out and to analyze and using your technology. That would be really cool. So I want to know your comments.
So what I would say is Richard's team and some of his colleagues at Lawrence Livermore that were participants in the innovation of the 280 nanometer, the very short wavelength non-visible light. One of the things in practice that we do differently that I think what you're describing is we use what by microscopic standards is a Grand Canyon of distance between the lighting, the way that we obliquely light the system and the tissue surface. And what that allows us to do is it compensates for the thicker slip covers and variability. Glass is not glass. There's a lot of people don't understand the complexities that slip covers have. 14 microns of variability in flatness. There's different levels of what I would call coarseness in glass. And there are a lot of things that you have to, when you're doing something at this level that has a global implication where you have suppliers from a number of different places that have to be addressed. And so applying just simply 280 nanometer UV light to a scander type, very close 2 millimeter working distance, that causes other physical problems with imaging, and you would lose a lot of the, I believe, spectral resolution that you would expect to see. And it would be, I think, degrading the integrity of the ultimate output, which we want to keep the fidelity as high as we can. So the concept is there, but in practice, it's a little more difficult to do. But obviously, if you could do that and adapt these types of technologies to things that were already kind of in place, I think that would be interesting. But it's not really our market. It's not where the resources we do have are focused right now. We really are trying to get out of the slide and out of the kind of very close operative distance, because I think there's a lot more to be had in the richness and fidelity of the data.
I will make this our last question in the interest of time, to be respectful. And the last question that I have on the screen is actually something I would love the audience here and the online audience to let us know through the Muse website, LinkedIn channel, wherever you want to communicate with us, or visit us at the booth 528. What excites you most about what we have just talked today? Like, what do you think would be the applications? Like, where do you see it? Oh, in my work, I would apply it here. Frozen section, efficiency workflows. We would love to hear from you. Thank you so much for joining us live and online, and thank you very much to my esteemed panelists. Matt, any last words?
Well, on behalf of Muse online and here in person, I'd just like to thank everybody for the time and learning about what we're doing. We're excited about bringing this into the market this year, and we're very privileged to have the colleagues that we have collaborating with us. And thank you to Richard and his colleagues for putting in the time to you know, I think a lot of people lose the forest from the trees. The settings at like Davis and Livermore, where there's a lot of grant time and money to develop these types of technologies, you know, I think commercializing them sometimes is the easy part. You know, we have a really good team at Muse to do that. The invention is the hard part, and so thank you, Richard, and the team for that.
I want to add to that. I'm going to try to get in 30 seconds if I can.
Go ahead, Jeff.
Is that? You know, after we did that big conversion in veterinary pathology to whole site imaging, a couple years later, I had a fear of missing out, so I went to the second annual World Congress on Digital Pathology to see what, especially mitotic counting, but also I was very interested in a talk by a gentleman I had not met at that point, and he sits 3 people to my immediate right, Dr. Levenson. So I sat in his lecture that day. I'll never forget where I was, the chair I was sitting in, because for decades I had thought about, there's got to be this better way. We're so archaic. You know, we lose samples. It takes forever to do this. The problems with histology laboratory. There's got to be this magic box where you dip the tissue in like something like DiffQuick and you drop it in a portal, and somehow it makes an image and a pathologist reads it. But that was just a will of the wisp, because there was no technology available to do that. And as you just said, Richard, technology drives science. And it would take 2 decades after that thought, when I sat in that lecture and I saw Dr. Levenson put up that slide of that canine mast cell tumor and then the fusion image of those orange cells sticking onto collagen, which does a very unique thing with mast cell tumors in the dog. And at that moment, it was, you know, the piloerector muscles in my arms contracted and the hair stood up on my arm. And, you know, I felt levitated slightly, the Levenson levitation, I guess. It was one of those moments I will never, ever forget. And, you know, in a single word, I was gobsmacked. And I was determined at that point that that technology is going to find its way into point of care veterinary hospitals, and we're ready to do that in my profession. We're right there at the cusp of it. So thank you, Dr. Levenson. It's a gigantic privilege to be, you're a legend for me.
Well, I just want to say that I've also learned over the years, well, thank you for that. That's really unexpected and a real pleasure, is that I've been in company development, and I've learned, coming from technology, that the actual point is, it's not the photons, it's the people. And these are great people. So I'll just leave it at that.
Thank you so much. It was a pleasure to be your host today. Enjoy the rest of your evening. And if you want to study this presentation, it's going to be available through Digital Pathology Place channels. Thank you so much.